Um, while we're waiting to get started, I just want to say good morning to Robert. Hi, Robert. Good to see you're on this thing. This is Joan Bailey Wilson talking. I am going to, I'll be on only for part of today, but I will be joining for the rest of the course, and I'll try to be back on later this afternoon. I have a bunch of meetings, but I just wanted to say good morning, and I'm so glad you're doing this class, and I'm really excited that it's going to be recorded so that future students and future users of S.A.G.E can use the software. So hi to everybody.
Thank you.
Yeah.
We'll be starting shortly. Thank you for coming.
Hello. Good morning, everybody. Yenju and I were here much earlier at 9:30, but we waited till we could see enough people turning up. So we're happy to see you all. First of all, I want you to know that Yenju Song and I will be presenting this S.A.G.E course. And we're giving it because we had a request from Wayne State University to give a virtual course. A couple of things to start off with. If we get disconnected for any reason, please wait. I promise you we'll come back, but I don't think we will, but just in case we do. Secondly, please feel free to ask any questions anytime. Keep yourself muted until you want to ask a question. And when you want to ask a question, unmute yourself. Also put your video on, because I will be able to understand you much better if I can see your mouth and lips. Okay, with that, let's start. Now, we're going to start off with give me 1 minute. We're talking about the basic principles of human genetics. All right. So first of all, genetics, this is very simple stuff, but we got to get everything straight. It's a science of inheritance. It's concerned with the differences and similarities of individuals resulting from the mechanisms of genetic material. It's the same root as generations, gene. That's the important thing. You have to have generations. Now, in genetics, we have a father that has the sperm and a mother that has the egg. The sperm has half of the father's genes. The mother has 1/2 of the mother's genes and cytoplasm. It's important to remember that. And then you get the fertilized egg with 2 sets of genes. And eventually, after the embryo, child, adolescent, you get the adult. Now, you have to remember there's an environment, and the environment starts with the mother's uterus. It doesn't start at birth. And then you have nutrition, infection, smoking, and such like. So the basic principles are as follows. The genotype is the genetic makeup, all the DNA received from the mother and father. And it's generally considered to be constant over time. The phenotype, on the other hand, is the appearance of the individual. What is measured? Okay, by the way, it helps to know some Greek, but that's okay. It's the pheno means appearance. For example, you might be observing a disease status, blood pressure, weight. And the important thing to remember is that the phenotype changes over time. It depends on both the genotype and the environment. And so the phenotype can be considered a function of the genotype and the environment. So what is the physical basis of the human genotype? So in every nucleated egg, a cell, you have genetic material in the nucleus. You have the DNA. Now in humans, we have 46 chromosomes, 23 from the father and 23 from the mother, 23 pairs of chromosomes. By the way, when I was a student many years ago, we had 48 because somebody miscounted. But we know now, and you've got to understand that everything I say from now on, I'm talking about the usual situation. Sometimes there are exceptions, but the usual situation is 23 pairs of chromosomes. And these are made up of 22 pairs of similar autosomes, homologous autosomes, and 1 pair of sex chromosomes, the X and the Y. And the XX is female and the XY is male. There are exceptions to this, but they are rare. The cytoplasm contains mitochondrial DNA, and it mustn't be forgotten. Now we have to understand the process of mitosis. This is the process of cell division to produce the somatic cells. We start off with the sperm, father's gamete, the egg, the mother's gamete. You get the fertilized egg, that's one cell, and that replicates and replicates until you get the adult. Each diploid daughter cell contains the exact replica of the 23 pairs of chromosomes. Meiosis. Now, meiosis is a very important concept. It's the process of cell division that produces gametes, sperm, eggs or sperm, by reducing the number of chromosomes from the diploid number of 46 to the haploid number of 23. Diploid means twice and haploid means once. So it's a haploid number of 23. What happens here is a bit complicated. First of all, the chromosomes, the homologous chromosomes, pair up together. And the next step is that they duplicate. Then you get crossover or recombination of 2 of the haploid forms. And you get what's known as reduction division 1 and reduction division 2. So in reduction division 2, you will see that there are haploid gametes. So let me make some different definitions. We're going to do a lot of definitions now. First of all, the gene. What is a gene? So using a modern definition, a gene is a DNA segment that codes for a functional unit, whether or not a protein is produced. Now, this is the definition. If you look up the paper by Gerstein 2007, I think that's a good definition. And it's made up of exons interspersed with introns. But you have to understand, a lot of people these days still think of gene as coding perhaps a protein. They don't say protein coding gene, but that's what they mean. But nowadays, you should think of the gene as a segment that codes any functional unit. Now, the locus is the location of the gene or genes on the chromosome. Now, what I want you to understand is that the word gene has changed in meaning. Even when I was a student, it had 2 or 3 meanings. The Drosophilists used to talk about 2 loci for each gene. Now, you'll understand why I say gene or genes in a minute. But it is so, for example, does the gene code for the ABO blood protein type, or Are there several genes there? In my day, there were 3 genes, A, B, and O. Now the molecular geneticists call it one gene. They call it the ABO gene. And that ABO gene is on the chromosome number 9. Alleles are different forms of a gene or different genes that occupy the same locus. So when the molecular geneticists say they are cloning genes, what they really mean is they're cloning alleles. And so this is very complicated because you have different types of geneticists using different kinds of words. So the alleles for the ABO blood locus are all found at a specific locus on chromosome number 9. Further definitions. Allelic genes are genes that occupy the same locus, whereas non-allelic genes, they're genes that occupy different loci. Linkage is the process by which alleles at different loci that are close together on the same single chromosome tend to stay together because the probability of a crossover is small. So let me talk about Mendelian segregation. Mendel's first law was the law of segregation. I think you probably all heard, you know, that Mendel was a monk and he did a lot of experiments on sweet peas in a town called Brno, which was then part of Czechoslovakia. Now, for a given locus, each parent transmits one allele, either the maternal allele from the mother or the paternal allele, in other words, the allele that parent got from his or her father, independently to each offspring, the zygote. So let us consider a single autosomal locus with 2 alleles, which we will call A1 and A2. There are 3 kinds of genotypes. There's the A1A1. This is called a homozygote, a homozygous for allele A1. There's A1A2, the heterozygote or heterozygous. And then there's the genotype A2A2, a homozygote, but now homozygous for A2. So if we look at this table, on the left-hand column, you see the parental genotypes. The first row talks about the genotype A1A1. That can give rise only to A1 alleles, and therefore the probability that the A1 is transmitted is 1. On the other hand, look at the next row, the parental A1A2 can give rise to either A1 or A2, each with probability of half, and A2A2 can give rise to gametes only that are A2. So the probability that the gamete is transmitted, the A2 gamete is transmitted, is 1. Now consider Mendelian segregation. Consider the mating type A1A2 by A1A2. So the gametes from one parent are A1 and A2, and the gametes from the other parent are A1, A2. One parent corresponds to the rows and one other parent corresponds to the columns. So if you multiply up and you see in this square that A1 occurs a quarter of the time, A1A2 a quarter of the time, A2A1 a quarter of the time, and A2A2 a quarter of the time. So usually, not always, but quite often we think of A1A2 as the same as A2A1, but we'll see exceptions to that later on. So the offspring genotypes are A1A1 with probability a quarter, A1A2 with probability a half, and A2A2 with probability a quarter. So in general, for a single autosomal locus with two alleles, which we'll call A1 and A2, there are three possible genotypes, A1A1, A1A2, and A2A2, and therefore 9 possible mating types, of which 6 are distinct when we ignore the sex of the parents. So here are the mating types, and we see what are the probability of the offspring genotype given the mating type in this table. In the first row, we see if we have a mating A1A1 by A1A1, the probability of the offspring is A1A1 is 1, and the other two are 0. The next row, if we have a mating type A1A1 by A1A2, the probabilities are a half and a half for A1A1 and A1A2, but the genotype A2A2 has probability 0 from this mating type. Then in the fourth row, we have these probabilities we've just seen, a quarter, a half, quarter. And if we have an A1, A2 by A2, A2 in the last but one row, we cannot have an A1, A1 offspring. That probability is 0. The other two probabilities are half each. And last row, if we have A2, A2 by A2, A2, then only A2, A2 offspring is produced with probability 1. Now this, I'm going to tell you my terminology, and it's important you understand. The probability that an offspring has genotype U, given the parents have genotypes UF and UM, that is the father and the mother have genotypes UF and UM, that probability is a transition probability. It's the probability of the offspring given the parental genotypes. It's a transition probability. And the probability that a parent with genotype U transmits allele U, which I call TOR, the U transmitting A, is a transmission probability. These two terms were first defined in a paper I wrote with Stewart in 1971. But you have to understand that a lot of people, a lot of writers mix these two up and they call them both transmission probabilities. But we have to consider them distinctly. Please remember that. So let us contrast the genotype with the phenotype. The genotype is specific genetic constitution of an organism, right? But usually the term is mostly used to pertain to specific traits or loci, i.e. we often use genotype to denote the alleles present at a particular locus, or we may be talking about two loci. The genotype is never observed directly. Let me stress that. Never, never. We, as far as I know, we still cannot actually observe genotypes. They are inferred through a phenotype. As far as I know, I may be wrong here, but the molecular people can correct me if I'm wrong, but I don't think it is possible yet to observe any genotypes. Only a phenotype, what you see, and what you see is not necessarily the genotype you think it is, or you hope it is. So the genotype is usually restricted to the DNA sequence and epigenetic phenomena. The phenotype, on the other, is the observable expression of the genotype and possibly the environment, possibly even some errors. Now, a very important concept is the phenoset. The phenoset of an individual's phenotype is the set of all genotypes consistent with that individual's phenotype. That term was first introduced, back in the 1940s, late 40s. Phenoset, a very important concept. We shall be using it quite a bit. So let's consider as an illustration the ABO blood group. Now on the left, you see 4 pictures, and they're the proteins in the membrane of a blood group A person, of a blood group B person, of a blood group O person, and of a blood group AB person. Now, if you look at it, you'll see that the blood group A, top left, has the A antigen. Blood group B next to it has the B antigen. Blood group O has neither antigen, and blood group AB has A and B antigens. So now let's consider the phenosets on the panel on the right. The phenotype for blood group A has genotype AA or AO. If you look at A and O, you can't distinguish which genotype you've got. So AA and AO is the phenoset of blood group A. Similarly, blood group B can be BB or BO. So BB and BO comprise the phenoset of blood group A. The AB, on the other hand, can only have genotype AB. The phenoset contains one type only, and similarly for O. So I hope you've understood the concept of a phenoset. It's all the genotypes that are consistent with a particular phenotype. Now let's consider dominance and recessiveness. So this is a very important concept, but unfortunately, we tend to talk very loosely. And for the purpose of doing any serious analysis, you have to be very clear as to what you mean by dominance and recessiveness. So classically, they refer to the situation in which the expression of one allele in a heterozygote masks the other at a particular locus. So the allele that is expressed in the phenotype is dominant with respect to that phenotype. The other one that is masked is recessive. Now, I stress with respect to that phenotype. Often you'll read and people won't even tell you the phenotype they're talking about. They think you automatically know they're talking about the disease, but you don't have to. Let us describe the relationship between phenotype and genotype. Okay.
Expressed in the phenotype is.
Was that a question? Was somebody talking? Not a question. All right, you decide to mute yourself. Yeah, please mute yourself until you want to ask a question. So, another term you will read a lot about is codominance. And all that means is that both alleles are expressed in the heterozygote, or that the heterozygote phenotypic expression differs from that of either homozygote. Now, some writers who should know better, they will say they will confuse codominance with additive. You must not confuse those two. Additive means the heterozygote is halfway between the homozygotes. Codominance just means it's different from either homozygote. So you can have overdominance, and that means the phenotypic expression of the heterozygote does not lie between those of the homozygotes at all, and that can occur. Let's continue with the relationship between genotype and phenotype. So let's consider a single autosomal locus with two alleles, which I call diallelic. A1 and A2. Now, I must warn you, most people now write biallelic. I just think that's bad English. If you know any Latin and Greek, you'll know that biallelic is a bastard word, half Latin, half Greek. So diallelic is completely Greek, all right? I have to admit, had a classical education. I learned Latin and Greek, so I like things to be correct. But most people these days, without realizing it, they're using the bastard word biallelic. So consider this diallelic locus with A1, A2. Dominant and recessive describe the relationship between alleles and, I stress again, the specific phenotype you're talking about. Sometimes it's assumed, usually, in fact, it's assumed without saying anything, but we've got to be very careful here. Consider the phenotype diseased, all right? Then you'll see the three genotypes on the left, and with respect to the diseased phenotype, if A1 is dominant to A2, then A1A1 is diseased, A1A2 is diseased, and A2 is normal. And not only is A1 dominant to A2, but if you have A1 recessive to A2, then the A1 is diseased and the other two are normal. Now, if I'd said with respect to lack of disease or unaffected, then it would all be the other way around. So you must remember what you and specify what you're talking about with respect to what phenotype when you talk about dominance or recessiveness at a locus. This will become clear if we look at sickle cell anemia. Sickle cell anemia is a very good example. There are three genotypes, capital S, two normal alleles. Capital little s is the sickle cell trait, often called the carrier because it carries the little s. And little s is the disease. You have the disease sickle cell anemia. Now, the disease is sickling of the red blood cells. The red blood cells, instead of being roundish and ovalish, they look sickle-shaped. All right. On the other hand, the trait is sickling of the red blood cells in low oxygen tension. So if you the trait says that it's only when you have low oxygen tension do you have sickling of the red blood cells. So little s is recessive with respect to disease, but dominant with respect to what we call the trait. So that is very important to realize. We always must specify what is the phenotype we are talking about. Continue with genotype and phenotype. The phenotype can vary among persons with the same genotype from no disease to variable disease degrees of severity. That's called expressivity in the literature. By the way, this literature is very much the kind of terms you'll see when people were studying only diseases. And this goes back to, maybe the 40s and 50s. Penetrance. Penetrant is the probability of disease given the genotype, because perhaps not everyone with the genotype will be affected. So for the above example, with complete penetrance, we have this table for the probability of disease given genotype. So this is called complete penetrance. We have A1 dominant, in which case any individual who has a genotype including an A1 is diseased, or A1 recessive, in which case only the A1 genotype has the disease. So let us continue with incomplete penetrance is a term that's often used. It refers to the failure of a phenotype to be evident, even though the genotype usually producing that phenotype is present. In other words, you really, it's not a one-to-one correspondence. And there are a certain probability that somebody who has the usual genotype for that disease, for some reason, doesn't show it. That's called incomplete penetrance. Variable age of onset, for example, Huntington's disease, also called age-dependent penetrance. We'll be talking a lot about age-dependent penetrance later on. Now, we'll use the term penetrance function in this course. It's used to describe the probability, mass or density. It could be, it doesn't have to be purely mass, it could be mass or density. It's the probability that an individual has a particular phenotype given a particular genotype. That's the penetrance function. Sporadic's a term that people use, or certainly used to use. Excuse me, I got to get rid of this. My telephone went off. Will you excuse me 1 minute while I take a call? I apologize for that. One of my worst fears was getting a call during this course. So we have the concept of sporadic, which refers to the presence of a phenotype, even though it's the genotype usually producing it is absent. So we have, oh, I know what I'm doing wrong. This is very complicated. Here we go. Variable expressivity refers to variation in the expression of a trait, for example, the severity, or more generally. Imprinting means the phenotype depends on the sex of the transmitting parent. So that's a situation where an A1, A2 person is not the same as an A2, A1 person. If we order the alleles according to the parent they received them from. Let us consider the word syntenic. Syntenic means if two loci are syntenic, they are on the same chromosome, or same chromosome pair, depending on how you, I mean, I think of a locus as being on basically on a chromosome pair, but some other people may think of it as just as one chromosome. We say two genes are on chromosome 9, but we usually, when we say chromosome 9, we mean the chromosome pair. Then there are two words you may see sometimes, paralogous. These are loci that arose from a common ancestor locus by duplication, and it's to be contrasted with orthologous. This is the same locus in different species which evolve differently. In other words, they differ by evolution. Here's a very good reference you might want to look at if you want to understand the difference between paralogous and orthologous. Pleiotropy. So pleiotropy is the occurrence of multiple diverse effects due to segregation at a single locus. The effects on multiple traits due to a single allele. For example, phenylketonuria, PKU. Phenylketonuria, it causes excess phenyl ketones in the urine, but it also, if untreated, will lead to a certain amount of mental retardation. So that's a pleiotropic effect. So if a single locus produces an effect on several traits, that locus is said to be pleiotropic or to have pleiotropic effects. Phenocopy, on the other hand, is a condition not genetically caused that mimics one that has a genetic component. For example, deafness. So there is segregation at a particular locus that causes deafness. It's recessive with respect to deafness. On the other hand, you may become deaf because of listening to loud noises, or these days, ordinary music bands and things. Then there is the thalidomide syndrome versus phocomelia. I suspect you are all too young to know what the thalidomide syndrome is. Well, back in the 60s in Europe, doctors were prescribing thalidomide for morning sickness when a woman was pregnant. And we're very lucky in this country that the Federal Drug Administration did not allow thalidomide come to this country, because what happened throughout Europe, there were a lot of births in which the limbs were not fully formed, either the legs or the arms. Whereas there is a genetic condition, phocomelia, and phoco means seal, the limbs look like the flippers of a seal. So that would be the thalidomide syndrome is a phenocopy. Genetic heterogeneity refers to the situation where different genotypes can produce phenotypes that are clinically indistinguishable. You've got to understand a lot of the genetics that was, developed during the last century was to do with clinical phenotypes. So you may have allelic heterogeneity, and most monogenic diseases have allelic heterogeneity. Or you can have, and that is to say that different alleles, can produce the same genotype, the same phenotype. Or you can have locus heterogeneity, and the classic example of this is elliptocytosis, where the red cells become elliptical. And this was shown back around, I'd say 50 years ago or so, that this so-called clinical condition, elliptocytosis, could be caused by segregation at two different loci. And you've got to understand this was at a time where we used to use linkage analysis, which we'll talk about later. And by linkage analysis, you can distinguish the segregation at different loci. What is epistasis? Epistasis is an interaction, that is, say, non-additivity between alleles situated at different loci. For example, where an allele at one locus masks or prevents the expression of an allele at a different locus, or otherwise affects the expression in a non-additive manner. Now, you have to understand that interaction. In general, statistical interaction of factors on a measured response depends on the scale of measurement. Whenever you see any interaction, you must ask yourself, what is the scale on which I am measuring the phenotype? For example, if you have an additive model, then multiplicative effects represent interaction. So you can think of epistasis as interlocus interaction between alleles at different loci, whereas dominance is intralocus interaction between alleles at the same locus. Note, dominance and epistasis depend on the scale used to measure the trait, because they are both kinds of interaction. Now, the effect of a binary trait may be measured on a penetrance scale, or it could be measured on a function of that, for example, the logit, the probit. And so you may or may not have dominance or epistasis, depending on how you measure a binary trait, what penetrance scale you use. Linkage. Linkage is the location of genetic loci sufficiently close together on a chromosome that they do not segregate independently. If two loci are close together, then the segregation of those two loci is not independent. Crossing over. Crossing over is a reciprocal event in a pair of homologous chromosomes. It takes place when the chromosomes have already duplicated at the beginning of the first meiotic division. Crossing over and the recombination fraction must not be confused. So crossing over says there's this crossing over taking place, but recombination, and in particular the recombination fraction, is the probability that an odd number of crossover events takes place between two loci. The recombination fraction is the proportion of recombinant gametes. Now look at this figure. You'll see on the left is when the two, you can think of this being a genotype, and when the chromosomes are, when things are lined up, as I showed some time ago, and if there is a recombination, then we change from we mix up the chromosomes, if you like. Look at on the left, the AA locus. We'll see that's yellow on the left, clear on the right for little a. The next locus, BB, on the right, we see little b, capital B, and I've colored it. So you can see there's an odd number of crossovers. So there has been a recombination. On the other hand, between the A locus and the C locus, you will see there has been an even number of recombinations, and even number of crossovers, and so there has been no recombination. Okay, so you have to distinguish the recombination fraction from crossing over. So recombination fraction is the proportion of the crossover, an odd number of crossovers gives rise to a recombination. So let us continue. Genetic distance. Genetic distance measures the amount of crossing over. On the other hand, there can be interference. Interference is a situation where crossing over in an interval influences the amount of crossing over in a nearby region. So we have map functions. Map functions relate the recombination fraction to genetic distance. And there are different kinds of map functions. We have the Haldane map function, which does not allow for interference. And there are a lot of other map functions, including, for example, the Kosambi map function, which is one of many map functions that allow for positive interference. So let us consider linkage disequilibrium. This is a 2x2 table showing the alleles. We have a large population of size n, and looking at the two locus genotypes, we see that little a individuals have allele 1 at locus 1 and allele 1 at locus 2. Little b individuals have allele 1 at locus 1, but allele 2 at locus 2. And then C is the number of individuals that have allele 1 at locus 2 and allele 2 at locus 1. And similarly, D is the number of individuals that have allele 2 at locus 2 and allele 2 locus 1. This is an ordinary 2x2 table. All right. And you see the margins. There's n total individuals, and they each have different numbers of alleles. Now, consider this. There is this quantity which is called D primed. First of all, let's consider D, not D primed, D. In the table, D is just AB minus BC. Okay, so you can see it's just the difference between the cross products. Now, in genetics, what we want to do is to standardize that in some way. So D primed is defined as D, this AB minus BC, but divided by its maximum value. And you've got all the details there of what its maximum value could be. So D primed is just a standardized D, where D is the difference between the cross products in the table. So let's see the next slide, next thing. The log of the odds ratio is just the logarithm of AD divided by BC, okay? That's the log of the odds ratio. And the correlation of the alleles in this table is equal to this D squared divided by the product of the margins. You'll recognize that as the usual chi-squared statistic, but divided by n. If you take the usual chi-squared statistic and divide it by n, you get R squared for these two loci. Linkage disequilibrium is a special case of gametic phase disequilibrium. And you need to understand this, that there's a difference between gametic phase disequilibrium and linkage disequilibrium. Now, let's consider the different kinds of traits we might have. First of all, the phenotypic traits of interest may be of various sorts. They could be discrete or continuous. They could be univariate or multivariate. And they could be, well, discrete traits, quantitative or qualitative, are categorical and can be dichotomous, sometimes called binary, or polytomous. Let's see some examples. For example, affected versus unaffected status. That's a binary node, or you might have no disease, mild disease, severe disease, and that is polytomous. Or you might have blood group designation. This is also a qualitative trait that is polytomous. On the other hand, continuous traits are always quantitative and are measured on at least an interval scale. For example, you may have measurements of blood pressure, anthropometric measures, lipid and lipoprotein levels, or you may have, whoops, oh, somebody just joined. I got a little ding in my earphones when somebody joined. Let's continue with this slide. Yunju? Okay. The phenotypic differences can be caused by allelic variation at a single locus. That's called usually monogenic. A few loci, that's called oligogenic, or at many loci, it's usually called polygenic. It would be more, to the point to call simple locus a unilocal one, few loci porsilocal, and many loci multilocal. As far as I know, there's only one other person who has suggested these three words, but we'll stick with what everybody says, which is perhaps not as good in at least my mind and one other person's mind, and we'll use the terms monogenic, oligogenic, and polygenic. If you have any of these plus the environment, you automatically have multifactorial, many factors. Multifactorial means determined by multiple genetic and or non-genetic factors. It just means many factors. Now, the term polygenic literally implies multiple factors that are exclusively genetic, but it's also used for a model involving the environment plus multiple genetic factors with a particular type of effect. For example, the simple additive polygenic model, or on a particular scale. Consider a single locus. In general, there can be k alleles at a locus in a population, but only 2 in any one individual. So suppose there are k alleles in the population, A1, A2, up to AK. Then the possible genotypes, the pairing of 2 alleles, are given in this table. You can see that there's A1 across the top, the 1, 2, 3 across the top, and A1 through AK along the columns in the across the side, the rows. And so you've got all the possible k squared pairing of 2 alleles. So the number of distinct homozygotes is the number down the diagonal. That's k. The number of distinct heterozygotes depends on whether you consider AI, AJ, and AJ, AI to be equivalent or not. If they're equivalent, which we often do, then the number is k times k minus 1 over 2. On the other hand, if they're not equivalent, you have k times k minus 1. So the total number of genotypes, if we consider the Aij to be the same as AJAI, is K, number down the diagonal, plus K times K minus 1 over 2, and that leads to K into K plus 1 divided by 2. That's the total number of genotypes at one locus when you have K alleles. Now let's consider the term allele frequency. In a population of n persons, each with 2 alleles at a particular locus, then the total number of alleles is 2n. So the relative frequency of the A1 allele is the number of A1 alleles divided by 2n. Now, every statistician knows that frequency is a count, but geneticists don't know that, and they call the relative frequency of allele A1, they call it the frequency. We talk about the frequencies of an allele when we really mean the relative frequency. So in a population, the allele frequencies can change from generation to generation for various causes. And these are, there can be abnormal locus segregation, what's known as segregation distortion, meiotic drive. All the genetic contributions are not equally probable. There can also be mutation. Another possibility is selection. Depends on the fertility of the parents and the viability of the offspring. And then there can be migration. And lastly, I say lastly, there can be chance, and occurs in small populations. You just don't have the frequency changes fast just because of this, the fact it's a small population, and this is known as genetic drift. So allele frequencies can also change with age within a generation and could be sex dependent. You have to realize that the allele frequencies are not fixed. So I'm now going to talk about Hardy-Weinberg proportions. I stress the word proportions. Consider one autosomal locus. Let us assume panmixia. Now, when we say panmixia, we typically assume lots of things. We assume normal locus segregation. We assume equal allele frequencies for males and females. We assume no migration, no mutation, selection or migration. And we assume a large population. Well, I apologize for that. I don't know what's going on with my computer. It started collecting more errors again. Anyway, if we have panmixia, then I take it you can all hear me. I hope you can. Yeah, okay. If we have panmixia, then random mating with respect to a genotype of interest, an individual has the same chance of mating with any other individual of the opposite sex. So the genotypes of mates are randomly paired. Now, if you look at the elementary textbooks on genetics, they go into a big hoo-ha about this, but it's very simple. You just have randomly paired genotypes, because if we go a bit further, we see that because the genotypes of parents are randomly paired and the parents randomly transmit one of their 2 alleles, there is just random pairing of alleles. And the genotype frequencies in a population depend only on the allele frequencies. Let me tell you a story. So Weinberg was a German physician, and he wrote on this maybe around, I don't know, 1908. Hardy was an English mathematician at Cambridge University, and we're now talking about the 1950s, I think, 40s or 50s. And one of his biological friends at dinner posed him this problem, and he just wrote the solution on his shirt cuff. And his biological colleague said, "You've got to publish this. This is so important, this Hardy-Weinberg stuff, you know." So being a mathematician, he was a little bit embarrassed. And this is, by the way, this story is absolutely true. And so he thought, I don't want my mathematical friends to see this. So he sent it across to Science to be published in the United States, where he hoped none of his colleagues would see it. So that's the story. Anyway, let's go on. For an autosomal locus, let QI be the frequency of the allele A1, and the genotype frequencies are said to be Hardy-Weinberg proportions. I'm going to stress the word proportions. When the genotype of AI-AJ has frequency A1, the frequency of AI has frequency QI squared, and the frequency of AI-AJ is 2QI QJ. And if you just have two alleles, A1, A2, with frequencies Q and 1 minus Q, then we see that there's three different genotypes, and the frequency or relative frequency of those genotypes, I use psi, the Greek letter psi, for the frequency or probability, really. Those three are Q squared, 2Q in times 1 minus Q, and 1 minus Q squared. So let's continue. You've got to realize this. Genotype frequencies at one autosomal locus attain Hardy-Weinberg proportions after a single generation of random mating, regardless of the genotype frequencies in the parents. That's if you have one autosomal locus. If allele frequencies do not change over generations, then they are in equilibrium. So far, we've only been talking about the proportions. If you have Hardy-Weinberg proportions plus equilibrium, you have Hardy-Weinberg equilibrium. So it's important to remember that it is possible for allele frequencies to change over generations, yet the genotype frequencies satisfy Hardy-Weinberg proportions in each generation. That was shown by Lee in 1976. So the point is, whenever people say they are testing for Hardy-Weinberg equilibrium, they are probably not. They've just got a random sample, one generation, and they're testing for Hardy-Weinberg proportions. You have to know that the gene frequency, the allele frequencies aren't changing from generation to generation to know that you have equilibrium. So take a break. What is the time now?
Right now it's 11:05. We were supposed to have a break from 11 to 11:10. So instead of like a 10-minute break, we can have a 5-minute break if you'd like.
That's what we'll do. We'll come back in 5 minutes. Okay.
Before we go, there is one question on the chat that from the AJMS. So what's the word segregate defined?
How is the word segregate defined? Segregate, of course, in this context, has nothing to do with segregation in the usual idea of, racial segregation. Nothing. This is a completely genetic word, and segregate just means that to segregate means to make differences. And so the alleles segregate, the alleles of a parent segregate when offspring are produced. In other words, You start off with somebody who has a genotype, let's say A1, A2, for those two alleles, and those two alleles segregate in the next generation. So it's very important to understand we're talking about the genetic term segregation. I'm not talking about any other term that you might be thinking of. Okay, thank you. And that was from Alexa. I recognize the initials. Thank you, Alexa. So now we'll start again in about, it's 11:07. Let's take 5-minute break, okay? 5-minute break now. Thank you.
Thank you.